Hi, Sergei from 100x Bio, and this is the third video where I'm introducing my path to starting 100x Bio as a company. And in the first video, I was discussing my school times, my university times, early times into PhD. In the second video, I was discussing like how I wrapped up my PhD and started a postdoc in immunology of cancer, and how I was discovering what is the best career path for me. And ended up thinking that becoming scientist in the industry will be the best path to potentially start my own company later on, or at least grow the ranks through the startups or pharma companies to have more roles which are more meaningful and have higher responsibility, because that was what I was looking for. And I would say with my extremely long PhD, like I spent 8 years before I was able to defend my thesis. And I didn't want to lose time. I wanted to move quickly because life goes. And so I figured that I want to become to continue my career as scientist into industry as R&D scientist. And I wanted to be somewhere in a space between discovery research and translational research. And I ended up exactly there. I started looking for jobs during COVID. It was extremely tough. And I found my job after a year of search. And it was very hard. What helped me is having a bunch of networking calls and having a lot of people's feedback on how I should look for jobs. So I recommend for every job seeker, like if you want to understand what is not working in your job seeking strategy, just talk to people, ask them questions, tell them what you're doing, and ask them what they would have done differently to make this process more efficient. And it took me 1 year to get an offer from Greenlight Biosciences as a scientist too in developing mRNA vaccines. It was Human Health Department, and it originated based on company's good position in RNA science in general, because they had long story of building RNA products for agriculture. And I can say nothing but just praises to Greenlight. Amazing company, amazing CEO, Andrei, and amazing experience. Even if we didn't make it, even if the Human Health Department eventually was closed because the capital markets were not like permissible to raise more money towards the vaccine projects, and I would say at this point there was such a strong and established competition looking at Moderna and Pfizer-BioNTech and some other vaccine companies, that like building a strong competitive vaccine pipeline, it would really be very complicated and very risky, I would say, also very risky. So what I'm leading to, I think Greenlight made an amazing attempt to create nice new vaccines, which would be like there were different value propositions, like to make them more affordable, to make them easier to transport, to make them affordable specifically in low-income countries and developing countries. And I really appreciated the mission of Greenlight. I really appreciated the culture of Greenlight, and I joined it to be responsible for T-cell responses measurement, because vaccine is inducing antibody responses, which is for vaccines against viruses is considered primary correlate of protection. Like if there is high titer of antibodies, we expect that someone is protected from infection. at least from severe infection. And with COVID, it was more and more appreciated that we should not only track the antibody responses, it's extremely important to track the T cell responses, because one thing is there are usually no antibody responses without the T cells. Most of antibody responses rely on T cells, and this helps you to justify the mechanism of developing of the antibody responses and confirming how vaccine is efficient. And then people may be interested to know how long-lasting is vaccine effect, and long-lasting presence of the T cells helps to maintain some sort of immune memory. And T cells themselves can protect from severe disease and death. This is very well shown for COVID and very well shown for COVID vaccination. And I joined primarily for studying the T cell responses. Then that team dynamics was slightly reorganized, and my main project was reactogenicity, like how our vaccine may be inducing some unwanted immune responses, not just shaped by adaptive responses, but also innate responses. And some of those innate responses can lead to adverse reactions like inflammation or high temperature or pain or whatever else we don't like after vaccination. And simultaneously, some of innate responses to vaccination may decrease efficacy of those vaccines, because early inflammatory response can suppress mRNA vaccine from being expressed as a protein. And if this inflammation is suppressing generation of a protein, we have less antigen and less response against this antigen. And due to this, there is no good protection after the vaccination. So we want to avoid what we don't like and generate something that we like. It's a complicated science. I don't want to go too far into this. But I was happy. I was really happy into the team. I was really happy with my projects. Probably first time in my whole life. I was happy with the pay, and like it was ideal job for me at its time. It was best in my life, and like I really appreciate those times. But at some point, it was clear that the company has challenges in financing the vaccine development projects, and at some point I started to look for alternative opportunities, and I was interviewed at a small startup which was developing new method for studying the T cells. I don't want to disclose the name in this video, but they were developing the method to study the T cells in a single cell format. So cells are like flying in a stream of tiny droplets. So tiny droplets, every droplet has on average one cell inside this droplet, and it is merged with another droplet, and T cell gets stimulated. And then we figure out that it's stimulated, and we start to sequence the T cell receptor, and now we know what target is stimulating what receptor sequence. And this is important knowledge for TCR discovery, T cell receptor discovery. And the discovery of those T cell receptor sequences may be important for generation different types of therapeutics. For example, like there are a lot of TCR-based therapeutics, therapies. So I don't want to go further into this, but I had a big question to a person who interviewed me. I asked them, like, we have huge variety of TCRs, huge variety of the sequences of these T cell receptors, and we have huge variety of the T cell receptor targets. So normally the T cell receptor is interacting with a complex of MHC, or in human it's called HLA with a peptide. And this peptide is this like non-self signature. signature of non-self protein, for example, or mutated protein. And when this peptide is presented, TCR is recognizing this peptide in a complex with MHC or in human HLA. So those peptides, they are built of several amino acids, and it's like up to 20 amino acids, and each amino acid in this sequence has, like, we have 20 amino acids in a 20 amino acid sequence. So it's 20 multiply 20 multiply 20. So it's a huge randomness, huge variety. And I asked them the question, like, how do we guess what T cell we stimulate with what peptide? And because we have 100 million, that's a range of a number, like 100 million of various TCRs in a person, and crazy trillions, 10 in crazy crazy trillions of peptides. We almost never guess, we almost never stimulate the T cell with the right target, and this brings us to the problem that most of the cells end up in waste, and like 99.99% of our sample is just thrown away. How much of a sample we can afford to use for this assay? And I didn't have any like good answer to this, and I was not moved forward into the interview process, but couple nights I could not sleep, thinking of like how complicated is the process of studying antigen-specific T cells, because after the vaccination, of course, we expect that we have expanded populations of T cells recognizing the vaccine antigen. But without vaccination, T cells are extremely rare. Okay, even after the vaccination, we have a long sequence of an antigen and some random presence of those non-self peptides which can be recognized by T cells. They're called T cell epitopes. And very often it's very hard to study where exactly within the antigen are those T cell epitopes. And T cell epitopes can be different for different people with different genetic background. So that type of study is extremely complex and expensive. If people want to collect all the data they need to know about precise immunogenicity of different pieces of antigen, T cell immunogenicity, they will get in trouble. Like the experimental process will be very expensive and very inefficient. And I started to think, okay, usually they're using the T cells from the tissue of an animal where animal is already vaccinated. So, okay, we have higher chances of finding larger populations of T cells, recognizing something associated with this antigen we use for vaccination. And simultaneously, we never stimulate them one by one. We stimulate them, we take at least 300,000 of those cells. And what do we treat them with? More often than not, instead of treating them with individual peptides, with individual targets, in vaccine development, people are treating them with just peptide pools or this antigen as a protein. So there can be multiple T cell responses involved into this study. And of course, information about precise epitopes within this antigen is useful, because when vaccines are built, scientists want to figure where they can cut it shorter to make the sequence more compact, or where they need to make it longer to maybe provide additional functions and maybe higher T cell response. So this knowledge is useful, but it's very hard to gain. And currently, the limitation of like finding rare T cells is solved by using a lot of T cells to start from, a lot of cells to start from, and a lot of targets pooled together to stimulate the T cells. And in some great examples, we can find like 0.5% of T cells recognize among 300 of peptides. And this is a good immune response. So if someone wants to study individual responses against any of those 300 peptides, it becomes more complicated. And I know there are different ways around for studying this. I want to explain that most mainstream idea, how this statistical problem is usually solved, many cells against many targets. And I was curious how to solve it more efficiently and how to find precise T cell responses. So I came to the idea, what if instead of treating the cells just once with a pool of peptides, to treat them with the peptide number one, with the target number one, and somehow count how many cells respond to target number one, and then remove it without losing any cells, somehow gently remove it and add target number two and record how many cells respond to target number two, and then remove target number two and run those cycles of T cell activation and data recording, data collection. And how exactly this is organized, I presented in the deck. I presented in different other communication channels. I don't want to stop here for too long, but I'm just explaining why I even started the company. Is because I found this interesting solution that no one else was using, and it seemed to be very simple to explain. And I started to talk to people. I interviewed 10 people from my close network. I posted on Facebook, guys, look, I have an interesting idea how to study the cells in the new super efficient way. And I interviewed 10 people, and all of them told me that I can patent and I should patent this. And some of them told me to build a company around it. So I thought, okay, why not? And this is how it has been started. And then I interviewed more people. I interviewed 30 people before I decided that I must test it in the lab. And then in the early beginning, I watched a series of videos, Y Combinator Startup School, YC Startup School, and it really helped me to solidify the idea that this project can be venture-backed. If I build a product, for this product, if I validate the demand and I prove very big efficiency gains, and one of efficiency gains comes just from automation. So this can be scaled up. And if there is thousands of the labs across the globe who might need it, this can be a venture-backable story. And I spent a lot of time talking to people who are prospective customers, prospective buyers of such equipment or such technology. And I've heard too much that it should be feasible. Because in the beginning, I didn't have any proof of concept. I was just asking them, do you think it's feasible to develop this method? And all of them told me, most of them told me, it seems to be feasible, and it seems that you can build a company around it, and you should try. And some of the people just told me, you're the person to make it. You're gonna make it. And people really encouraged me to start a company. So I wanted to rent some space, some lab space, and I figured that I need to incorporate first. So I incorporated, and throughout my entrepreneurial journey, I was using a lot of advice from YC Startup School, and so far I applied to YC 4 times, and I'm planning to apply more, and until I get there. So that's a plan. And and I think it was great advice to start testing and building as soon as possible. And speed is extremely important. And now I feel that I somehow lost the speed of development which I had in the beginning. And I think that it's due to some lack of experience but I'm trying my best to maintain good speed. So I rented the lab space, tested the hypothesis, and the main key idea of cycling uh, worked in my hands, and the cycles were short. It is important to run multiple cycles. You need each cycle to be short to be able to run a lot of them. So the concept was running about 100 cycles in less than 20 hours, about 16 hours, each cycle taking 10 minutes. And can you imagine, it's more than 1 year since I started my first experiment. I still was unable to test more than 20 cycles because like building the prototype is something that is not very easy. And there are many tiny technical details which you never know before you try to build it. What's next? Next, I had to file a patent, so I worked with IP law firm, which has like 20 years of experience patenting in this field, and they helped me to file a provisional patent, and they told me, Sergei, you can pay us after you raise some money, which was extremely useful for me at that point because I didn't have my personal money to pay for the patent filing. And then after Greenlight, Human Health Department and Greenlight was closed, I started full time at 100x Bio. And I started from raising money from friends from Facebook, which is another funny story. I had a blog on Facebook. You can call it a blog or just regular posting, and we have some sort of community of people interested into evidence-based medicine, into technology, into let's say innovation, or just discussing the news, discussing the memes. I think that our community is mostly like Russian immigrants to different countries, and after a series of posts about me building a company because I wanted to share with people what I'm doing, it was so exciting. I was looking for people to discuss what I'm doing. Some people just suggested me in the comments that they would invest some sort of money, and I figured how to collect those small early investments, and this was extremely helpful for me to start building a company. And between August and late October, I raised already 140,000, and this helped me a lot. But in parallel, this misled me. This speed of fundraise misled me to think that it's going to be so simple to keep raising. And since then, I only raised 60 more thousand from individual investors again, but now not from people who knew me on Facebook for years, now mostly from people who just met me, and more often than not, they need to have some time to meet me better. And now, since I'm in the middle of my pre-seed and I need to build more connection with angels, accredited investors, which are very rare people, I need to be better understood. I need to develop the trust somehow quicker. I need to accelerate that trust building experience. And this is one of the motivators for me to start this video series, because like I can't spend hours and hours and hours with people to explain to them why what I'm doing, I believe it can be huge, why what I'm building, I believe it's going to work. Because for many people who invest into technology, they invest into technology developed in universities, and this is technology which already has millions invested, and it has labor of scientific teams working on those projects for years. They have tons of data. They have patents filed with assistance from the universities. They didn't have to spend their own money. They didn't have to spend investors' money. And then later on, they only need to license, and which is actually not that easy thing to do. But they have different types of problems than I have. And for investors, they would be like, you don't have any data. So far, I ran only 20 experiments. They like, and out of those 20 experiments, amount of data that I'm ready to show and brag about is like, it's like tiny fraction, because many experiments just don't work. It is normal. And the type of problems that I'm solving right now is complicated. So this video series are dedicated, at least in part, to trying to build the trust that I don't know myself. I did not know at all if this technology would be working before I ran proof of concept experiments. And now I think it's like 99.9% de-risked, but so far I don't have a nice data package to show even to the angels. And I need to find accredited angel investors who will understand the line of thought, like why it is 99.9% or even higher probability that it works as it should. And I have some proof of concept data. And I also have some papers, some studies of other people that I can show to them and explain like why I have no doubt that it's going to work. But in the end of the day, it takes some sort of leap of faith for these people to trust me with their money. And I want to present to you that I don't feel like I'm a crazy person. I think I'm doing reasonable things. However, I need to act in a way that can seem to be slightly crazy. Like, I need to make some bold decisions and operate with lack of funds, operate with tiny chances of finding the investors who invest in this profile of a company. So having all this said, I still believe that I'll find the right people. And I rely on some sort of viral effect. I rely on the effect of, the handshake connections, like we are connected to so many people through 1 or 2 or 3 handshakes. I want to be seen and heard right now because I'm looking for very rare people. My method is intended to find very rare cells. My outreach now is intended to find very rare people who will understand the potential upside, who will understand that making some kind of crazy moves, I still maintain some sort of sanity, and I have an ambition to build a successful company. And if this like 0.1% risk still realizes, I have some list of like backup plans. Because it's not that I'm stuck into this technology, stuck into developing this technology. I want to develop a line of products. I want to develop a viable business. I want to develop a viable business that not only builds life science tools, but also builds the diagnostic platform, maybe builds some proprietary line of therapeutics. So I more or less understand what the venture capital people want, and I more or less understand what person profiles they see as a perfect partner for the long-term journey. And it's not ideal for them to invest 2 million and return 10, which is like 5x return. It is cooler for them to put 20 million and return 40, right? Which is 2x return, but they make 20 million in this case. So I understand that throughout my career, starting from the small steps, I can scale up later, and I think that I can later on learn how to run bigger scale projects. So that's my career goal. And I hope that this reasoning is presented in a clear enough manner, and I want to find my people. I want to find my people who will want to work with me. So I'm wrapping up. I will try never to run videos longer than 30 minutes, but this case. This video was long. Thank you for watching. And you know whom I'm looking for. If you know those people, please connect me with them. Send me the link. Send them the link to this video, or send them the link to my LinkedIn profile, because the technology that I'm building will help us develop quicker and cheaper, develop better vaccines, develop new immune therapies against cancer, against autoimmune disease, develop new modalities of diagnostics. I'll be talking more about this later, and here I'm wrapping up the story of 100x Bio creation. This is how and why I created this company. Thanks for watching. Bye bye.